In this video, we're going to talk about strategies to determine when you have an Sn1, Sn2, E1, or E2 reaction. And so this really involves using this chart that I've got shown here. This is also posted to, so I think I shared the wrong window. So this is the chart, and this chart is posted to Canvas. Now let's actually bring up the notes movie, or the notes screen. Okay, so we have the notes in front of us. So I just kind of pasted the chart that is uploaded to Canvas in Module 6. And this is the reactivity chart is what this is called. And what this allows us to do is look at the reaction conditions and determine which type of reaction we expect to see. In Sn1, which is always coupled with E1, and Sn2, which is separate then from E2 reactions. And so to use this chart, what we have to do is just understand the axes, and then once we have this chart in hand and we have our axes understood, we can just kind of correlate the type of reactant as we see. Let me circle that for us. We have our substrate and then the type of additive to that substrate. We can categorize that additive as as being a poor or a good or a poor, I guess, nucleophile or strong or weak base. So let's start with the substrate. So the substrate, that's our haloalkane. And the haloalkane can be either methyl, primary, secondary, or tertiary. So what we do is if we see some sort of reaction here, plus something, if we're going to predict what type of reaction it is, so predict the type of reaction, we see that this is a primary haloalkane. And so we would focus on this row of the chart. I'm going to uncircle that. So that's the primary row. Okay, so that's how you use the substrate row, just kind of categorizing things as methyl, primary, secondary, tertiary. This is something we should be okay at, but what about the additive? And what we do is we look at the basicity and determine if something is a strong versus a weak base, along with the nucleophilicity, which is good versus poor. I like to use instead of strong versus weak. Okay, so we've got these various categories, and what I'm going to do for the rest of the time is just use the chart that's in front of me. Maybe you want to have a chart that's in front of you from that separate window as I talk about each of the columns. So again, the rows are the substrate, which is the haloalkane, and then the columns, which we have to understand, are the additive, which is the nucleophile or the base. What I want to start with is the poor nucleophile, strong base scenario. So again, these two attributes for our additive are the basicity and the nucleophilicity. So we need to understand something about both of these aspects. That is how nucleophilic the thing is, how basic it is. So it's a poor nucleophile and it's strong base. This is the last category. And these are our E2 bases. E2 bases. Okay. So E2 bases are these two guys. And they are poor nucleophiles because they're too hindered. But they're strong bases. And the reason why I know that is because they have negative charge on N or O. If you've got a negative charge on an N or an O, then you've got a strong base. Now, are there other strong bases out there? Yes. But what I'm trying to do is just give you something to work with that you can apply to a number of cases. Yes, there are more strong bases out here. These aren't the only two examples. But what I'm saying is when you have a negative charge on an N or an O, you can assume that it's in the strong base category. Now, if it's an N or an O with these big things hanging out, it would be a strong base and a poor nucleophile. It's too sterically hindered. What if you have something else? What if you have something like this, where it's just an ethyl group? This would be an example of something that has an O minus, so a strong base, but it's not an E2 base. Now, the key thing about E2 bases is that they are strong bases and poor nucleophiles. Here, this is a strong base, but it's a good nucleophile. We won't worry about kind of medium nucleophiles or something like that. We're going to try to keep it as black and white as we can. Just using this chart is already impressive enough. So this would be an example of a strong base and a good nucleophile. And that would be the third column from the right, or excuse me, third column from the left. So strong base, good nucleophile, these would be O minus and N minus without being an E2 base. Are there other E2 bases? Sure. Okay, but we're not going to worry about it. We're going to worry about just the two E2 bases that I've given you, which if I zoom out are just those two things that are highlighted in green. So what we've done is we've covered half of the table by just understanding that if an O minus and an N minus is present, we have a strong base. Okay? What I'm going to do is flip the notes now, and we're going to look at the rest of the chart. So what about, there are some exceptions to the O minus, N minus strong base, and they are N3 minus, this is azide, is a weak base. And then not really an exception, but just one to pay attention to is that Cn minus, which is cyanide, the negative is on carbon. And so this is also a weak base. Both of these are good nucleophiles, and we'll look at poor nucleophiles here in just a second. But this would be two examples of good nucleophiles and weak bases, which is the second column. The second column. Okay, now this is actually the biggest category. Okay, this could be things like I minus, I mentioned Cn minus, N3 minus, just trying to think of, I guess I'm blanking on the examples, but these are the examples that don't fit the other three categories. And this is a very large category. Maybe I put PPH3 in here, for example. So I'm just trying to think of some other examples that I've given us. These are kind of the big ones, I guess. So that's the second column. And now this is the biggest group. So you might want to highlight this or just make note that this is the biggest group, and you're going to get to this category by either recognizing cyanide, azide, those two that I just talked about, or really anything that doesn't fit the other three categories. Okay, then the last category is, I guess, the first column, and that is poor nucleophile, weak base. Now these, for this class, are just going to be water, which is H2O, and alcohol, which is ROH. Okay, what we see is that this is ROH. There's no negative on the oxygen, which means it's not basic, and it's not a good nucleophile. Okay. Let's go ahead now and break down each of these columns. Now that we know what we can expect each of the things to be to lead us to each of these columns, let's go through and kind of look at the various examples. So what I want to start with is that first column. So first column, which is poor nucleophile and weak base. For these, you could just put a note here that if you see something attached to an alcohol or H2O, these fit in the category of poor nucleophile, weak base. That's going to lead you to the first column. And then from there, you look at the haloalkane. We have methyl, primary, secondary, tertiary. Now, if I just look at the table, it has the letters NR for the first examples, and then Sn1 E1 for secondary and tertiary. What I want to point out about this column is it's a bit awkward. In the primary and secondary case, we have Sn1 E1 reactions. These are our Solvolysis reactions. So in Solvolysis reaction, we would have something like this. Now, if you notice, this is a tertiary haloalkane. And we would react that with ethanol. Ethanol is an ROH, which means it's a poor nucleophile and a weak base. And we would see Sn1 E1 reactions. And I know this is not the easiest reaction we've talked about this semester, but it's one we've gone over before. So I won't go through it in incredible detail because we've done it before. But this is the outcome. And so we would say this is a Solvolysis reaction. You have to add the ethanol, then lose the ethanol. You actually have to use the ethanol as a bulk solvent for this reaction to go, but otherwise it doesn't work. What we want to notice about this reaction is that it requires a secondary or tertiary haloalkane. Otherwise, the reaction fails. Okay, so the reaction fails if you don't have a secondary or tertiary haloalkane, and the reason why is because the carbocation won't form. Now we see that effect of the reaction failing because the carbocation won't form. If we look at the first two entries of the column, we see no reaction for methyl, no reaction for primary, and that's indicated with the letters NR. And the reason why is because the carbocation fails to form. It's not until you add more substituents to stabilize the carbocation intermediate, that is the secondary and tertiary examples that you actually see a reaction occur. So that's the first column broken down into the reaction types that we would observe. Okay, so the second column, again, this is the broadest column in terms of the types of things. I guess I should move this over. I'm going to first have my haloalkane. So we have haloalkane, then we have good nucleophile, weak base. These are typically things that are negatively charged or that have nucleophilic lone pairs of electrons, but we don't see them pick up hydrogens as easily as a typical O- would. So we again have methyl haloalkanes, primary, secondary, and tertiary. These are going to be, these broad cases were in many times Sn2 examples. And as a result, we're going to see Sn2 reactions at methyl, at primary, and at secondary. In tertiary, though, we're not going to see Sn2 examples, but we could see Sn1 examples. And if we're making carbocations by way of an Sn1 process, we're also going to see an E1 process occur. Now, in some secondary haloalkanes, we're going to see competition between Sn1, Sn2, and E1 processes. So we either have Sn2 or we're forming carbocations to give rise to Sn1, E1 products. This is kind of the least useful entry. This is not something I plan to ask you about on the exams. I would say I won't, but then I accidentally did on one exam ask you about something I said I wasn't going to ask you about. Anyway, okay, my plan is not to, so call me out if I do. But for the secondary ones, it's a little bit confusing to know which one you're going to get, Sn2 or Sn1 E1. It's just one of those things you have to go into the lab and actually mix the chemicals together to see what happens. But this is kind of our Sn2 category. The second column, these are the Sn2 reactions. Okay. Now the third category is an example of when this table gets to be particularly useful because it allows us to see a switch between reactivity. So I just want to remind us before I write the chart here that good nucleophiles / strong bases, our third column, these are RO- and R, let's just put O- and N-, but not E2 bases. Okay? So with that in mind, what we should notice about these is that the reaction actually changes. So for our haloalkane and our good nuke, which is also a strong base, okay, what we want to do is recognize that there's a switch. If we have methyl or primary, we're going to see Sn2 reactions in both cases. However, if we go to secondary or tertiary, the added steric hindrance of the additional substituents is going to steer us towards E2 reactions. And E2 reactions are going to take over, even though we don't have E2 bases. Okay, so that's a bit unusual. So we see things change. These typically we would see do Sn2 reactions, but with secondary now we're going to see E2 reactions, which means we have to be kind of careful when we're talking about inversion of configuration. There are probably examples where I wanted to practice inversion of configuration in Sn2 reactions and I accidentally grabbed something that was too strong of a base. I should have stuck with like N3 or Cn-. Anyway, moving on to the last column, poor nucleophile strong base. These again are our E2 bases. Poor nucleophile because we have the added steric hindrance. Strong base because we still have O- and N-. And as a result, we're going to see mostly E2 reactions. That is, if we've got our poor nucleophile strong base, our E2 base, with the various haloalkanes, and maybe you're peeking at the chart and you already see something interesting. But for primary, secondary, and tertiary, our E2 bases are going to tend us towards E2 reactions, whereas the methyl-containing ones actually, interestingly, divert reactivity towards Sn2 reactions. But the important thing to note is that a methyl haloalkane cannot form elimination products. Now, forming elimination products relied on taking a vicinal hydrogen and forming a carbon-carbon double bond. If you've got CH3Br, you can't form a CC double bond, and there are no vicinal H atoms. And as a result, you're going to see no E2 or E1 for that matter, or E1. So only Sn2. Now I want to show you an example of that. If we take our E2 base like this and react it with bromomethane, what happens is that once we cross out the potassium and draw a negative charge, that negative charge will undergo backside attack. And you might say, well, Willoughby, didn't we design this E2 base with enough steric hindrance so that it couldn't do an Sn2 reaction? And the answer is yes, we did. However, in this case, the substrate, the haloalkane, is so small that an Sn2 reaction is actually viable with an E2 base. And we're going to see addition of a methyl group, a CH3 group, to our oxygen with a negative charge. Okay, so if we look at practice exams that use this chart, you can see what I'm kind of going after. In old exams that were done in person, we would I would provide the chart for the students, and they would, instead of asking them to memorize it, and then they would just show that they could apply it and recognize substrates and that sort of thing. So I expect something similar will be asked of you all. So hopefully this was helpful.